Good evening, everyone. Welcome back to Weekend Chat with Researcher, Session 3, Season 2. We are Bugbear groups at NCBS, and this is our outreach channel. Through these sessions, we aim to bring research closer to science enthusiasts with the belief that anyone sitting at home can understand science and research irrespective of their academic background. I'm Akshara, and Nitish will be the co host for this session today. Hello, Nitish.
Hello. Hello everyone, and welcome again. So uh, today, um, like let's discuss what are we. Let's talk about what our session will be is going to be. So Akshara, in our past sessions, like in our first season and this season also, we talked about some small organisms which are like quite microscopic, right? Or you can say we talked about microorganisms, and we came to know that they are everywhere in world, like including our body, uh, like E. coli or lactobacillus, which reside in our body, which aids in digestion. And there are microbes which are pathogenic to us. And like apart from us, even plants also that you see around, they also have microbes associated with them.
Interesting. So, do these microbes harm them or help them? Uh,
Like I'm not actually quite sure about whether they harm them or they help them. But to learn about these microbes, whether which are these microbes and whether they actually harm these plants or not, we have our guest, Dr. Pratibha Sanjenbam, uh, who is a postdoc fellow at Deepa Agash's lab, and she did her master's and PhD from VIT. And in her doctoral work, she worked on isolation and identification of candidal compound, so from Streptomyces, which is also a bacteria. And after her PhD, following which she was a research associate at VIT and a guest faculty at Kuvempu University, Shimoga, Karnataka. So with this, I would like to welcome Pratibha. So hello, Pratibha.
Hi, Pratibha. Yeah.
Pratibha, you are on mute.
Sorry. Thank you for the nice introduction. And today I will be talking into a very interesting world of mine, which I'm very interested in learning more about these plant-associated microbes. And hopefully I can deliver some of the basic fundamentals, how microbes associate with them and what kind of interaction happens between plants and microbes. Um,
Yeah. So, to begin with, you can share your slide.
yeah.
Yeah. To begin with, yeah,
Yeah.
what are the kinds of microbes that are associated with plants? Like there is a particular class, like only bacteria, or there are multiple different like classes of microbes that are associated with these plants.
Well, when we talk about plant microbes, it's quite diverse. It range from bacteria, it range from a small organism from which is the unicellular microbes to the multicellular microbes like algae. And so there are like it is quite vast. It can be bacteria, it can be fungi, it can be clusters of microbes together, which forms the plant microbiome as such. And with this, we also see that plant in general has these distinct parts. We have the underground parts, which is the root, and it's commonly known as the rhizosphere, where the bacteria or the microbes resides, and the phylosphere, which is the aerial part, which is I'll talk to you in later. So,
Okay.
Akshara, to your question, it's quite diverse. We don't have a distinct set of bacteria which resides in the plant. It can be a huge diversity which coexist together, and that community itself can impact the plant's growth, or it can be harmful as well.
Yeah, because we mostly relate these microbes to pathogens, right?
Yes.
So do these like help, like what most of them are pathogens, or like what, like do they help and harm them? Like what is the scenario there?
To answer that question, it's kind of 50-50, because
Okay.
sometimes if the pathogens load are really high, then the plants are being killed.
Okay.
They are infected continuously by the population of those pathogens. If the beneficial bacteria load are really high, then the plants can be more fitter. They
Okay.
can be more healthy. They can provide stress, resistance environment to the plant. So it depends on what kind of environment they are and what kind of microbes coexist together, and that impact might lead to whether it can be beneficial or it can be a pathogenic interaction.
Okay.
So the most, for example, if you see in the rhizosphere, we know this term from the beginning. The history started when Lawrence Hiller, the first scientist who coined the term rhizosphere. He was very curious to know why nodules were formed in leguminous plants, and he showed that this rhizobium can fix nitrogen for the plant, and the bacteria in turn gets photosynthetes, which are sugars, which are nutrient to them. So this kind of beneficial interaction would lead the plant to a better fitness. And when it interacts to a harmful environment where we have a very pathogenic bacteria load higher, then it suffers from any disease or infection.
Okay.
Yeah, so
So
it's like humans also, right? In E.
Yes,
coli,
yes,
beneficial, and sometimes we have travelers diarrhea
correct.
from that.
Sometimes we do even see helicopylori.
hmm,
Helicopylori to some, I mean, it exists in the stomach, but it causes some ulcers to some patient. But it may be neutral, or it can be just a commensal there in the stomach without causing any harmful effect. When we talk about these terms, then we can see this sort of different kind of interactions, in any host associated bacteria.
so you talked about nodules. So all these rhizosphere associated with bacteria form nodules, or there are like what, like do they always form nodules in the roots?
No. So the nodules are specifically formed in terms of these leguminous plant. They are very specific to legumes. But there are bacteria, certain specific bacteria, which can do the same function, which can fix nitrogen from atmosphere, but they are well associated with non-leguminous plants as well. Like
Okay.
for example, azotobacter,
Yeah.
which I will talk to you later, in in when we cross about these different parts and different association of these microbes with the plants.
Mm, yeah. So, so you talked about different kinds of association. Can you, what are these different kinds of association?
Yeah, so these associations are very common. It can be like, this is not restricted to plant always. It can be in animal versus microbes as well. So this can be classified into three broader classes. The first one is called mutualism, where both the partners, wherein the host as well as the bacteria or the microbe gets benefited each other. So for this, we have a very commonly known example, which is called rhizobium, which is found associated with legumes, and it is quite well known that this bacteria resides near these inside these nodules, and they fix air, nitrogen from the air, and in turn they get photosynthetes, or the sugars which is being produced by the plants as nutrients. So such type of interaction is called mutualism, and there is a very cool picture where we show this nodules form in a medicago plant, which is a leguminous plant, and inside this, the bacterial cells are found, which is rhizobium. So such type of interaction is commonly found in nature. We also have one such kind second class of interaction, which is called commensalism, where the bacteria or the where one gets benefited while the other is really unharmed. So this is very common. Like very, there are neutral bacteria which get nutrients from the host, but they don't do any full harmful effects on the host. And the third one, which is much more interesting, is again the parasitism, where the this is a common example of pathogens, where the pathogens get nutrients from the host, but they also harm the host. For example, the
Okay.
Erwinia, which is a bacterial group, they causes root rot disease in most of the plants. Then Fusarium and Rhizotonia falls to fungus family, but they also form this root rot disease, which is similar to this disease which is shown in the picture. As you can see, the roots
Uh,
are all infected and they turn black in color. So they become dead. They cannot do any more function, and the plant at the end dies due to the infection. So this is the three broader classifications
oh.
that we can make of interaction with plant microbes.
So we
So
have a
like,
question from Harini.
Mhm.
What do these microbes do to help the plant?
So, as I said, Harini, there are different types of microbes which can do multiple functions. One example is here, which is Rhizobium. They can fix nitrogen from the air. So, the atmospheric nitrogen, they can convert it into ammonia, and that ammonia can be utilized by the plant host as, and they can convert it into amino acids and for the synthesis of nucleotides and stuff, for the metabolism. So this kind of advantages they can have from the microbes.
yeah, so I was wondering, like, not all bacteria infect humans, not all bacteria infect plants. So, is there any something special about these bacteria which infect only plants, or do we exactly know that what classify a bacteria as a uh,
That's a very interesting question, it is, and it's still under, like, we are exploring a lot more, because microbes are diverse and they have certain specific signatures that they some bacteria can form like pili like of structure on the outside covering, where they can attach and adhere inside the plant body cells. Then plant also evolved to recognize this type of structures by their membrane kinases. So depending on what type of bacteria, it's hard to explain that this kind of bacteria will only associate with plant or will only be harmful to the plant, because how microorganisms, they keep on evolving and changing their uh, because this
Yes.
is an arms race, one has to survive. So the plant also adopt many strategies to identify these signatures, and the bacteria also tries to fight back by modifying their structures, or to invade inside. So it's very hard, but there are, like, for example, if we say that Rhizobium, there may be Rhizobium which may be bad. There are species that can be bad to the plant as well. It doesn't mean that when we say Rhizobium, it's going to be good for everyone. So it depends on what type of plant system and what kind of interaction it happen with the host plant.
yeah, so like as I remember from my textbook knowledge, these leguminous plants are like peas and beans, right? Or
Yes, soybean. Then we have this medicago plant.
yeah,
These are
so
all leguminous plants. Yeah.
so it's interesting that like what we eat in day-to-day life is like assisted by bacteria also,
Yes.
and
Yes.
yeah,
So now the another interesting part is we were pretty much focused in rhizosphere. Currently, we are now also exploring a part of plant which is the phylosphere. This is much more interesting because uh, why we are interesting in because before going to that, I'll just want to explain what is Phylosphere. Phylosphere is the aerial part of the plant. The entire body that is above the ground is called Phylosphere, and the leaf surface is called the phylloplane. So leaf surface is the one where many microbes are again found, like you do in the rhizosphere. So it's like a niche for a certain set of microbes. And it's very interesting because you have these abiotic factors, which is always in key role, right? The environmental factors like drought, then we have raining season, then we have this UV radiation from the sun. In spite of that, there are certain sets of microbes which resides there happily, and they are enjoying their community there. So it's very interesting that in Phylosphere also we found a very unique nitrogen fixing bacteria by Runin, and she termed the Phylosphere in 1961. So it was like very, then the study began that not only Rhizosphere, the Phylosphere has also a distinct role, and this is much more interesting that once we connect how this Phylosphere community can also linked with this Rhizosphere, how this plant balances body signal is much more interesting and unexplored area. I mean, people are exploring, still exploring right now.
Yeah.
So, like it's quite evident that these organisms are quite microscopic to our eyes, and we cannot see them. Like I personally did not imagine that small different world exists in front of my eyes, and I'm not able to see. So
Yes.
how do this, like for example, like first scientist to coin the term phylosphere, like how these scientists exactly found that there are some bacteria associated with plant. How do they do it?
So she found it in well, she was working in the Indonesian forest vegetation.
Hmm.
She found through a scanning electron microscopy that there were clumps of bacterial cells which were tangling around the leaf surface. But not going into the microscope, we can now easily do a very uh, a short very interesting technique which is very easy one. We just take the leaf and take an pressed on the agar medium, which is the food for the bacteria,
Hmm.
and you incubate it for some time. Then you have these microbes grown in the structure of the leaf.
Oh,
So yeah, so it's very interesting that we can see that these bacteria are in the shape of the leaf where you have pressed them.
yeah.
So people in higher studies can do, of course, do go for scanning electron microscopy and check what kind of association, what kind of microbes are present on the leaf surface. But not going into that, we can still find large number of culturable bacteria which can be grown in these bacterial plates by just pressing the leaf on those agar plates, which
Like so, like we with limited resources in colleges or
yes,
schools, even
yes,
we can find out
yeah,
whether there is some bacteria or not.
yes.
So yeah, so with this, like with this picture now, I like you got to know that there are some microbes on these leaves. So how do you identify? How do you know that this is Rhizobium or some other bacteria?
Oh, that's that's a basic biology. Once we get these colonies on the, these are called bacterial colonies. We purify them because now you can see they are all in mixture form, and that mixture form is called consortia. So that consortia we need to break down and make it into pure culture. So for that we do a basic microbiology technique which is called streaking. So we take the loopful culture, for example, the colonies from here, and then we restrict it. And then once we get a pure culture without any mixing of color or without any mixing of colonies, we do a DNA sequence identification, which is called 16S rRNA or rDNA amplification. And then we can find out what kind of genus or what kind of a species they are. In terms of fungus, you can do 18S, well as in case of bacteria, we can do 16S sequencing. So you have a short fragment of DNA, and then you can sequence that out, and you can know what kind of genera they are and what kind of species they belong to.
Yeah, so like to our viewers, like in our last session also, when we were discussing about phages, uh, our guest speaker talked about sequencing to identify phage and
Yeah,
to annotate phage.
yeah,
And so sequencing is another, I'll reiterate it, it's a technique where you get to know what the sequence of your DNA is, which is
yes,
the
yes,
genetic material.
yeah.
And you can kind of get to know
Hmm.
which bacteria or which organism exists in your study.
Yes. And it's not all the entire genome, but you can have a short fragment of the DNA which belongs to 16S and you can identify it easily.
Okay. So we have an interesting question from Harini.
Yeah.
Do the microbes travel from place to come to the plant, or it is that the plants like they are born with it?
Some are inborn, they because they reside inside, but some are like stochastically or randomly they colonize from the air or from the
Okay.
soil, from the environment. So what's happened is that plant also selects some bacteria given what kind of interaction they have. For example, when you produce flavonoids compound, then rhizobium bacteria are attracted towards the root. So
Okay.
similarly, some kind of signals is conducted between the host and the bacteria, and then the colonization happens near the rhizosphere or possibly in the phylosphere. That's how the colonization happen. And then the bacteria which are able to invade inside can reside as the endophytes, which are called bacteria which are residing inside the plant, and they can be transferred maternally through seeds, so they are there inside
Okay.
all the time.
Okay. Interesting.
Yeah.
Yeah. Um, so
So
I had a question. So like rhizosphere bacteria, they form nodules and stuff. Do these do these phylosphere bacteria also form some kind of structure, or they're just present on the surface of the leaf?
no, as far as the current knowledge or current studies, there has no been such report forming nodules on the leaf. But there are bacteria, for example, here, which is called azotobacter, was found on the leaf phylosphere, which can fix nitrogen.
Okay.
So they can fix atmospheric nitrogen as rhizobium does, and they convert into the ammonia, which can again the plant can assimilate easily as a nitrogen source for any metabolism or any such kind of
Yeah.
functions. So this kind of bacteria is found, but as far as the nodule is concerned, there is no structure which has been found so far on the leaf surface. Yeah. So we also, I mean, I also do a lot of leaf surface exploration because I am fascinated by plants because they are sessile organism. They don't move, but how is this signaling happens? Like, how did they choose these good microbes and bad microbes?
Yeah.
So for that, I use rice plant as my model system. So this is a piece of rice leaf, and as you can see, these are the trichomes, which is called the hairy structures. When you touch the rice leaf, when you there will be something like a tiny leaf hair structure. So that's are called trichomes. And what I found through scanning electron microscope was a diverse set of microbes, which was found on the leaf surface. Not only bacteria. So this is a trichome stalk. We have this trichome stalk in electron scanning microscope. You see these bacteria are adhering and attaching over here. And interestingly, when you see go to the sides of the leaves, then also you see a lot of bacteria adhering to it. And not only bacteria, we can also see a bacterial fungus association, which is much more interesting. You can see the filament over here, which is the fungal filament, and you see these bacterial cells attaching to it. So some kind of crosstalk may be happening, or maybe
Yeah.
some neutral connectivity maybe, but that was very interesting to see that even the rice leaves host a different set of microbes. We even found diatoms on the leaf surface, which is algae, most probably found in water condition. But these rice are grown in a wetland cultivation, which is where the rice has been like 3 to 4 centimeter of water
Yeah.
has been kept. So it was quite interesting to me that this kind of communities exist, in rice as well.
Exactly, like all classes of almost all, all classes of microbes are
Yeah,
present on the
yeah,
rice
yeah,
leaf.
yeah.
Interesting.
It will be
Pla-
fun to even understand the function of each microbes. Some may be neutral, of course, some may be pathogenic, of course, but it will be quite intriguing, and it's a very fascinating world how this in a small niche you have these different sets of communities living together.
Yeah.
This is the scanning electron microscopy image,
Yes,
right?
yes, yes.
Uh, nice.
So we have a question from a viewer.
Yeah.
Pooja wanted to know, can this method be used for other plants also, like mangroves, like the same method of identification of
Yes, I mean, what method she's trying to say? Is it a leaf pressing thing,
yeah,
or?
yeah, yeah.
Yes, we can still work on that. But however, we have to remember that what kind of bacteria are we interested in? Like for example, certain bacteria uses glucose. Like select, we can compose a selective medium, and when we press the leaf on that agar, we can still isolate that particular bacteria. Or if we are interested in a community, that will be little hard because fastages bacteria will grow very fast. They will over dominate the culture plate, whereas when you give a selective media, then it will only choose for that bacteria or a certain group of bacteria to grow it. So certainly it will work, but you need to know what kind and what type of bacteria you are looking at. And also there are a lot of fungal community, as I have said, mentioned already, that might dominate over the plate. So you might add some fungal compound in your plate, so that will work perfectly fine.
Okay.
And
So,
once you have, yeah,
continue, continue.
so once you have these colonies, then you can restrict it to get the pure culture and then sequence the 16 as fragment.
So, in continuation to Pooja's question, so do you expect different bacterial species, different plants like mangroves differently, rise differently,
Oh,
and
you mean different leaves had different communities?
no, different types of bacteria in different types of plants?
Yes, yes. Uh, so for example, like for example, in a given particular plant type, we see one particular bacteria X.
Mhm.
So maybe similar or different species might occur with another plant. So it
And
might not be necessary that whatever bacteria species that we get in mango plant might be occurring in the another plant. So they must be falling into the same genus, but they may have different functions or they may belong to different species.
mhm.
So
And what dictates that? Like what kind of controls that?
so many reports or studies recently says that the host plant itself selects it, have a selection. So as I mentioned, so if there are certain key molecules which has been secreted by mango, and then this bacteria X is only capable of using those or attracted to those exudates, then they are mostly thought to be dominant in that mango plant. Whereas in some plant where some key molecules are secreted, and then a certain group of bacteria that fall into the same genus might also show up in different plant. So it depends on what kind of interaction between the host and the bacteria have, and and that can have selection over the bacteria as well.
Okay, so we have one question from Harini again. So she's asking that could the microbes which harm plants whether they can harm humans? If yes or no, then why?
Okay. uh, Microbes that can harm plants can harm humans as
Humans.
well. uh, there can, I mean, there is not much report where we see that plants pathogens are harmful to humans. But certainly there might be case, Harini, but I'm not sure to mention it right now, that what kind of pathogens might be, because there are few cases which I have seen where foodborne pathogens infect, but those are all foodborne diseases. I'm not sure whether they are plant pathogen or they are uh, transferred during food preparation and stuff. So
Okay.
yeah.
Right. So there's like a lot of things to explore in this
Yes,
field.
yes. um,
Great. So how do these bacteria like exactly help, or like can you give some example of your work also?
yeah, so So as I mentioned that this Phylosphere is a very recently, what to say, going on work where we are trying to uncover what kind of mechanism, what kind of interaction, what kind of fitness benefit, or what kind of detrimental effects it might have on the plant's health. One such example that I found was very interesting. Like you can see, this is an experiment done in a lab. You have Arabidopsis plant over here. Exenic plant means sterile plant, so they don't have any microbes in it. They are made sterile, and then you infect them with a pathogen which is called Pseudomonas, which they have written as PST. So Pseudomonas strains, one strain, particular strain has been infected, and you can see how the disease progress during time. So 5 days DPI means after 5 days of inoculation, 5 days of infection, 12 days of infection, and 19 days of infection. As you can see, the plants have started to turn brown and they are dying. Well, you spray a Phylosphere bacteria, which is called Spingomonas, you can see they are much similar as the control plant, which is not infected with uh, Pseudomonas. That itself suggests that even Phylosphere occurring plants, these microbes can, give greater advantage to resist pathogens and to protect plants from the pathogens. So this is one such example. One of the interesting model system that I'm working right now is called Methylobacterium. So they are very fascinating. They are very uh, beautiful bacteria. They are pink in color. And the reason why they are predominant in the Phylosphere is that plant, when they grow during these stages, when they grow during their developmental stages, they have a very unique process which is called cell wall rearrangement, called pectin dimethyl esterification process. And that during this process, what happens is the cell wall, if you see a seedling time lapse video, or you see that the cells grow, I mean they keep on growing, so the cell wall undergoes expansion and rearrangement, and during this process the leaf or whatever the seedlings produces methanol as the byproduct, and this methanol has been taken up, shown to taken up by this beautiful bacteria, Methylobacterium, and in turn it has been shown to reside near the stomata, and it's many papers it has been shown to produce growth factors like auxin, which is a plant hormone, cytokinin, and also vitamin B12, which can enhance the plant fitness. So some sort of mutualism between the host and the bacteria. So right now in Deepa's lab, what I'm trying to look at is, okay, given this mutualism, such a relationship, what kind of interaction it might happen between the plant. So to look at that, what I'm trying to use is a local rice varieties, which are called rice landraces, which are very distinct, and these rice landraces are traditional rice varieties, so they are grown in a particular niche for several generations. So the idea is that if they have been grown in that particular area, they might have selection on their own bacteria. So what I'm trying to look at is each land race or each variety have its own Methylobacterium. Are they unique to each other? So it's the same question that someone asked from the group, like Pooja asked, right? Whether different plant have different bacteria or they will have similar bacteria. So right now I'm testing whether the different rice variety have similar Methylobacterium or they are different Methylobacterium. Is that distinct to each other? Distinction. So for that, what I have been doing is I have collected many samples from Northeast, because Northeast has hundreds of rice varieties in, because and they are very distinct to each other. They have local communities which has grown a specific rice variety for several generations. And these are the sampling locations where I travel to collect all these rice leaf samples. And what I do is, as I mentioned, they are very easy. You just need to know what kind of media you have to compose to get that particular bacteria. For my particular bacteria, Methylobacterium, as I mentioned, they use methanol as the carbon source. So what I do is I prepare plates with 0.5% methanol, and then you just take the imprint of the leaf, press it for 5 minutes, keep it there, then remove the leaf, incubate it, and then you have this nice beautiful leaf structure, as you can see over here in the plate, and then you purify it to get pure cultures, and then you can identify it later on. So in doing so, what I also do is I do not have access to lab, because these are all in the local communities. I don't have So for that I need to have a sterile environment. So we constructed this UV portable UV box, which will if people have worked in the microbiology lab, you know how this laminar hood works. The only drawback is I don't have airflow there, but you can do the
Okay.
imprinting quickly, and then you can incubate the plates, and then you start traveling again. So this is
Yeah.
how I do in the field. And
Yeah,
yeah.
interesting. So there's a question regarding this leaf imprinting method from Pooja.
Hmm.
So can this method be used for other parts of the plant, for example, roots or stem?
Roots, basically, I have not tried, but roots, you can always do crushing, and then you can do serial dilution. But for the stem parts, definitely it works. The only tricky part with stem is that all the bacteria clump together, because there is no proper venation or structure there. So all the
Okay.
bacteria, clump together, and they are very bit difficult to separate out.
Yeah.
So
Yeah, given the
that's
surface area is very less as
Yes, yes.
compared to leaf.
Yeah, yeah.
Okay.
So it will
Oh,
definitely work out.
okay.
So this is what I do in the lab. Uh, and what we are trying to know is this: given a population in rice variety of Methylobacterium, we try to ask what determines the distribution and the phenotypic diversity. So this is the same set what we try to distinguish whether the bacteria is distinct or not. The So what we have done is we have given different carbon sources, and we try to map out what are, so if they are associated with landrace 1, do they have certain set of carbon use profile? So in landrace 2, do you have certain set of carbon use profile? So that testing we have done in the lab, and it's now published. If people who are interested in can always read this paper. What we found that was, given a two landrace distinct, they are colonized by distinct bacteria with different choices of carbon source. Of course, methanol being common, because this is this is why they reside in the phylosphere. So this answers Pooja's question, that it is not necessary to have similar bacteria in different plant, but they might be falling into same genus, but they can have they can be clearly distinct with each other in terms
Yeah.
of function. uh, yeah, so this is one of the question that we are trying, and it seems like there seems to be landrace specific selection. So now we are trying to test whether Methylobacteria confer fitness. Is there any advantage in the field condition? We know that in lab condition, it's uh, it's 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 not replicable in the field, and that's not the exact scenario where the bacteria does in the natural environment. So we are now testing that whether bacteria can be useful in the field condition, and this is what we are doing right now. I mean, we have done in 2 years back, but we still analyzing the data, and we sought to see that, you know uh, if Methylobacterium is beneficial, then we should see that the yield will be much more improved in the rice grain. And that's what we are aiming for in future applications, to understand the natural ecosystem and to replicate in the field condition for future agriculture purpose. So this is where I am right now in the lab.
Okay. So you earlier you said that because there is rhizosphere, there is phylosphere. So do you find any kind of interaction between, like you've come across any study that show that there is any kind of interaction with these two spheres?
Yeah, so that's very interesting, and that's one of my interests in future, to
Okay.
understand how these two parts are being balanced by the plant.
Okay.
But there are no such studies, because it's quite challenging. Most of the bacteria that we find in the plant, we are just answering through the culturable bacteria, which can be cultured in the lab. but there
Okay.
are certain bacteria which are again unculturable. So with the help of this new technology, which is in the invent of omics, now we have know how to do whole genome sequencing, we know how to do metagenomes. With this integration together, we can now know what kind of networking happens between the rhizosphere and the phylosphere, and how the plant even signaling happen between these two.
Okay. Your the Methylobacterium is, so you have found out that it is beneficial. Like, and like, what kind of benefits does it provide to the rice plant?
So in my study, I found that in one rice variety, it was increasing the yield. We
Okay.
could not see any vegetative growth advantage because
Okay.
maybe the natural environment already has the other bacteria which can support them from the nutrients or depletion or stuff like that.
Okay.
Whereas what interesting is, we found that there was an advantage in the yield, and we also found that the OTUs of this pathogen load was also really decreased. So we are speculating right now
Okay.
that it might be advantageous for a biotic stress. It might provide protection from the biotic stress, and in turn the plant invest all the energy into the yield production.
Okay.
So this is what we find, but it is not true for all the land races. It's not, for some variety it doesn't have any effect. So it might be that the relation is not very strong for some land races, and it
Okay.
might be true that some land races have a strong effect and they depend on Methylobacterium.
Okay.
And future study needs to address some of this
Yeah.
underlying mechanism between the host and the bacteria.
So earlier you had classification of the kind of interaction, mutualism and
Right.
commensally. So what kind of interaction? So all the varieties have same kind of interactions, or like they're different? Rice variety have different kinds of interactions with these bacteria.
Yeah, so I am wouldn't comment that because we have not screened enough.
Okay.
It needs more screening whether they have these plant growth promoting traits, which are because we have not looked at how this. I mean, if what if they are capable of producing auxin, cytokinin, and stuff, which are reported in the literature. So my guess will be right now to that they might have a mutualistic kind of relationship, but it needs to be tested to conclude that whether there is commensalism or mutualism.
Okay.
But however, the literature suggests that there might be mutualism, but I mean, in my study, I have not tested yet, and it needs to be tested.
Okay.
So, like we know that in for like there has been good study about rhizobium, right? And it helps in nitrogen fixation and helps plant. So, can we like provide rhizobium from external, like extra rhizobium or
Yeah,
like
yeah, yeah,
rhizobium?
yeah, Nitish. Rhizobium is helpful when it is a leguminous plant.
Mhm.
So that it was a long back. People have already introduced this nigresin, which is a rhizobium fertilizer. So many people use it for supplementing it, and it's a good procedure rather using a chemical fertilizer, where you enrich the rhizobium as a biofertilizer, and then uh, you
Okay.
make the plant more healthy or more growth. So that's been in practice for a long time. Like, I mean, rhizobium is quite well known, the mechanism is well known, we know what genes get triggered, which genes are important for the nitrogen fixing. So uh, yeah, but there are a lot more which needs to be unraveled, because most of the model system in plants were arabidopsis, soybean, and we have, I mean, tons of plant species which are being not explored yet, and maybe there might have some interesting candidate as well, which we can use for our own bioprospecting in terms of
Mhm.
medicine, in terms of agriculture. uh, yeah, it can be useful in many ways. Not only useful, but it can be also interesting to know how this ecosystem, unique niche, even operate in the natural environment.
Okay. So we have a question from Harini. Can these microbes affect stem in a good way or in a bad way?
Yes, they can. You mean the bad guys?
Like, do they affect stem? So basically, like we like phyllo, we are mostly like we discussed about the leaf.
Yes, they
So
can even, I mean, they can still colonize the stem surface. They can even colonize the flower. I mean, the whole entire microbiology which deals with the upper part is called Phylosphere. But since
okay,
the leaf has more area, you have more microbes there. But still, the microbes, the good guy as well as the bad guy, can still colonize the stem or any parts of
okay,
the plant.
okay. Eh,
Yeah,
mm,
so like, so I was looking at the slide. So this illustration is made by Pranjal. So he, like, I follow him on Instagram. So he Sai art. Uh,
Yeah, yeah.
so that's also like an kind of emerging area where people use artist art to explain science, and so this
Yes.
has been like beautifully demonstrated.
Yes,
yeah.
it's really helpful to even, I mean, rather than giving um complicated graphs, it's very, it's very uh, interesting to depict our own results in the form of cartoons, and so that we connect with other people who are not into science as such, but we can still connect. To the other people as well. So I find it really interesting rather than, I mean, putting
Yeah.
difficult graphs probably.
So
Yeah.
just to give an idea of like how research happens in this plant field, and like we have Dr. Shiv Prasad in our campus also who works on plants. So as I know that these plants have huge lifespan, right? And like microbes, you can culture it and maybe it can grow maximum within weeks or maximum within weeks, right? Mostly in hours and in days. So but how do you as a researcher uh, deal with this lifespan related issues, because otherwise it will take years.
uh, so depending on the plant variety, it might depend on years, right? I mean, for rice variety, for rice plant as such, we need a duration of, depending on what kind of assays you put up, it may last from 2 weeks to four months.
Okay.
So but still the bacteria can divide, as you know, they can still colonize. That's the most interesting bit, that bacteria keep on dividing it. And there is no, what to say, when the host plant is still alive, there is no decline in the population of bacteria. We might see some decline during the time lag, I mean, when we monitor time to time, but there is no uh, limitation that the bacteria will get extinct or they will get exhausted, uh, because they are wonderful bugs. They can
Mm.
keep dividing it.
Yeah.
So
So,
with
Methylobacterium. So there was a question whether these plants are these bacterias are like they do the plant acquire or do they are born with it. So can you comment on this Methylobacterium, whether the plant is born with it or they acquire it?
uh, so that testing is still not proof yet, but they are also found inside the seed.
Okay.
So generally, they are metallally, I mean, we can hypothesize that they are metallally transferred, but
Yeah.
they are certain, I mean, they are still environmentally acquired as well, but acquired become as a partner, like how they are transferred through seeds is still not established or not investigated yet. So we still have a lot of options and lot of studies that needs to be conducted, in lab and in natural field condition, how this environment, Methylobacterium can be whether we is maternally transferred, or at what stage this certain species colonize,
Yeah.
and how they become partner from the plant. So that's still a mystery, because plants are always exposed in environment where there is lots of microbes, and still we need to be tested out.
Okay, great. So, like, we have another question from. Oh, yeah, we just got another question. First, from Harini, like, as we discussed about Methylobacterium,
Mm.
do we know that any plant is getting harmed by Methylobacterium?
Uh, yes, there are no such reports which suggest that there is Methylobacterium is harmful. But there are some certain cases and reports where Methylobacterium found in water was reported as a harmful pathogen for humans. Uh, but in plant, it has not seen as a harmful case so far. But in my study, one of the bacteria was found to be harmful, and we are investigating now, like what strain was it, uh, and uh, and what kind of factor it has that it is playing as a harmful role. So maybe some species or some strain of Methylobacterium might be pathogenic, and some might be a very good person. So this is true in every population. In a given population, we will always have a good person and a bad person. So that's true in bacteria as well. So we will have some strain which are very beneficial, which are some strain which are pathogenic.
Yeah.
So we have another question from Pooja, who has asked a bit of technical question. So if we don't have 16S rRNA uh, facility, like we don't have facility to sequence that. So is there any alternate method which we can use for identification of these bacterial species?
So as I mentioned, for Methylobacterium, it's quite distinct, and you can just do by a culturable technique. But other bacteria which are very similar in their phenotype, the colonies are very similar. For example, if you check some yeasty-like colony, you will also see that Rhizobium also form those kind of coloration, like what to say, that creamy colony like type.
Yeah.
So in that type of colonies, you might not be able to distinguish properly. So I am not coming up with any technique apart from 16S, because we need to sequence it anyhow to confirm it. Because
But
sometimes
broad
there are.
classification we can maybe do based on seeing the morphology of the colony or
Yes,
doing
to
some
some
kind of.
extent, yes. But then I'm suspecting for those phenotypes which are very similar on the plate. So that will be very difficult to even call out whether those are E. coli, or whether those are some Pseudomonas strain. But if we have a certain selective media where we know that this certain bacteria can grow in that medium, whereas the other cannot, then you can probably screen them by growing those bacteria in that particular specific medium.
Yeah.
Okay, great. So we are coming towards the end of the session, and I just have one last question. So, if we grow a plant from its seed and in a completely bacteria-free, isolated environment, right? So, do we see, or is there any study which sees that there's some kind of effect on plant growth? Technically, what I mean is like there's no microbe which can come from surround.
Okay. Uh, so you mean to say when a plant is grown in a sterile environment,
Yeah.
whether they get any benefit?
Or
Benefit or disadvantage on its growth, any kind of effect which we see.
No, absolutely the plant won't die. They will still grow, but when you have these microbes as a partner, then the partnership, that health wise they get improved.
okay.
So it is not what to say, uh, strict that the plant microbes need to be there to survive, but uh, when you have microbes, then the immune system is boost, then the nitrogen fixation is done much more easier than you do it yourself, invest more energy yourself. But the plant will still survive in the sterile environment, it won't die.
Mhm. And are there like specific classes of bacteria which change when through life stages of these plants, like when it's a seedling or when it's an embryo?
Yeah, certain studies have now started following up the assembly of bacteria. Like they are finding some particular bacteria in certain stage. For example, in the seedling stage, they find much more of a bacteria which can help in nutrient, aggregation and stuff. Where in the later on stages, they find bacteria which can provide resistance to biotic stress. To this is like I mean, with the event of new techniques, now people are trying to understand how this association is affecting throughout the life stages. And in the coming years, we will have much more closer look like what controls this assembly, and what kind of association happens at what stage of a particular plant variety, or is it true for a particular plant variety, or is it true for all. So that can be answered very easily.
Great. So with this, we finish our session, and we have a couple more questions. Pooja is asking which labs give the facility of 16S RNA in India.
Oh, we have, I mean, certain companies where we do
Yeah.
sequencing outside, outsourcing outside. You can send the DNA extracted from the bacteria and send them. And I mean, they will have, of course, you have to pay the price, the charges they have per sample. But this can be done. There are a lot of companies in India like Chromo Biotech, and I mean, many other companies, so that you can easily send your sample and get your sequence done. um, yeah, I think our NGS also do outsourcing, right?
Yeah. So like institutes like IASC also, I think NCBS
Yeah,
also. Uh,
yeah. So there are
they,
many companies and institute, Puja. You can, I mean, I mean, write to me or write to Nitish. You can, I mean, of course, get help from us, and you can send the DNA easily and find out what bacteria there are.
okay. And another last question from Harini. Uh, can these microbes harm seeds before even the plant arrives?
Yes, there are a lot of fungus which we have still problematic so far in seeds where they start harming. I mean, you could have seen in the rice hucks this black powdery stuff which is caused by aspergillus. Um, so yeah, these bad guys and are everywhere, like so they can infect seeds as well before we start germinating it.
Okay, great. Then with this, we end our session. And in this session, like it had been very informative and quite understanding session in which we got to know that there are good bacteria, bad bacteria, which do nothing. They just stay, and then and there's like bacteria interaction, bacteria fungal interaction inside a plant. So like everybody is interacting with each
With
other
each
in this
other.
wonderful way.
Yes. And that's how wonderful they are, right? This community itself.
Yeah.
I mean, it's fascinating. I mean, and because of some X and Y imbalance, we have this pathogen and we have this beneficial term. So
Okay, so with this, we end, and we would like to thank Pratibha for joining us today.
thank you so much. I have really a very interactive session today, and yeah.
Okay, so with this, we thank our viewers who joined today. And as you all know, this is our outreach channel, science outreach channel. And to join us, we have posted our mailing list link to keep yourself updated on coming sessions. And if you want to join our outreach initiative, do share our videos and increase our reach to science enthusiasts. And we'll keep learning about this wonderful nature around us and how we as researchers kind of explore or unravel the complexities of around. So Akshara, should we say
Yeah.
bye?
Yeah.
Okay,
Thank you,
great.
Pratibha.
Thank
Thank you
you.
all.
Thank you all. See
Bye.
you